Hi, everybody. I'm Monica. I'm a research scientist at the research center over there. That's Melissa. She is my mycorrhizal person. She did a master's with me, and now she's working as a technician. So I promised her I wouldn't make her talk, but you can ask her questions about her thesis.
Don't talk.
Yes, you. But yeah, I brought her along because she probably knows more about mycorrhizas at this point than I do, because she's been fully integrated into mycorrhizal stuff for about 3+ years, working directly on this stuff. So I have a little handout. So with mycorrhizas, unfortunately, this isn't like a hands-on activity. So, I mean, I threatened I could do like an interpretive dance or something, but these things you can't see, right? We can't dig these up and look at these. They're microscopic. So we thought about bringing a microscope out, and we didn't. So it's also up here. The picture's up here as well. Sorry.
Oh, good. Try to find the perfect spot so you won't get
So I won't get the feedback. So you're all looking at a little picture of what a mycorrhiza does. Mycorrhiza is a soil fungi that is absolutely dependent on a growing plant to get its carbon. So that's what I tried to illustrate there for you. The fungi is only ever growing when the plant is growing. And one of the things that it can do for your plants is once it colonizes the roots, it grows hyphae. It takes that carbon from the plant, and then it grows hyphae, and it can extend further and deeper than the plant root system to access nutrients. So it can be responsible for up to 100% of a plant's phosphorus, and about 20% of nitrogen is what we are currently saying, although those numbers change as research develops. So that's fundamentally what it is. It also holds on to more water, so it can increase drought resistance. So why aren't we all using mycorrhiza all the time? They naturally occur in the soil. There's lower diversity and lower biomass of mycorrhizas, usually under conventional soils, because we tend to fertilize a lot. If you fertilize kind of at the recommended rate, you are selecting against mycorrhizas, and that's primarily why mycorrhizas is not a biotechnology or a technology that we're applying currently in the field. So we can demonstrate that there's biomass and yield benefits, drought benefits, and then like nutrient uptake benefits. Melissa has when you get a chance, come stroll past. We just grew some winter wheat with and without mycorrhiza. And you can see, the wind's blowing away some of the roots, but she's weighed them, so rest assured that mycorrhiza has more biomass in the root and in the above ground. At the 8-day stage and at the 4.5-week stage is what we have. So yeah, it's not a replicated, it's just a demonstration. We just grew a bunch of these for you so you could have a look to see. But the key for this is to reduce your nutrients, right? Which is why it's not really something that people are open to using necessarily. And then the other reason why it's not a technology that we're using quite yet is if you go through the literature, you can see really good examples of like biomass and yield increases with mycorrhiza. And even when the conditions are great, when you're like fertilizing at a slightly lower rate, when you're doing all the things that are supporting mycorrhizas, sometimes it doesn't work anyways. And I mean, I would say generally we don't know why, and I think that's where a lot of the research needs to happen. I kind of think it's finding the strains that are going to match the genetics of the plant that you're using, and then ultimately like coating it onto the seed as you do with other microbes. I think that's where we're heading. But we're certainly not there yet. And part of the reason we're not there yet is because these suckers are a pain in the butt to grow. Melissa's nodding. I've been asking her to grow stuff for us to just do experiments. And it has to be associated with the plant to increase the biomass, right? So we have to be growing plants to be growing these things. Or we have to have roots in culture, essentially, to be growing these things. So it's really expensive to produce them. So ultimately, when people ask, like, should I be using mycorrhizas, I mostly say, well, it's a management thing that you can do, right? You can make decisions that are supportive of mycorrhizas, but not necessarily, I wouldn't say inoculum is at a stage that you can just use and expect good results at this stage yet. So we have, this isn't the best picture, but if you I'll shut up in a sec and I'll get you guys to ask me questions. But one of my PhD students that defended this spring did a fantastic field experiment with mycorrhizas and spring wheat in this case. And you can actually see with a half recommended rate of NPK and mycorrhiza, you can see that it's as good, basically as good as a 1x NPK, right? But he applied a commercial inoculum essentially, and it worked. Now, I don't remember which varieties of wheat, but it worked excellent with one variety, and there's no effect in a different variety. And I think that really goes with, like, you have to find the right match. And what's available commercially is like 1 or 2 or 3 strains that they're able to grow really, really well and sell, right? So, sorry to the companies. Hopefully nobody's here from the mycorrhizal company. But I think there's lots of opportunity, essentially, to find strains that are going to work with particular varieties. And I think there's a lot of potential there because then you can reduce your nutrients, which saves you money, right? And ultimately could be better for the environment because you're not losing all of that to leaching and all of those things. So I wanted to talk about mycorrhizal networks as well. So something that Melissa's been doing is doing these mycorrhizal network experiments. A thing I haven't talked about yet is that mycorrhizas are implicated in disease resistance. Melissa did her master's on ascochyta blight and chickpea. And one of the things that she found, and you can jump in and yell at me if I'm misrepresenting your work at any point. We grew chickpeas in these H pots in the greenhouse. And these come apart, and we put like a mesh in here that separates, that can basically keep roots from going through, but allows the mycorrhizal hyphae to go through. The mycorrhiza can actually connect 2 plants at the same time. So 2 living plants can be connected at the same time. There's a handout. Okay, thanks. Another handout. This is another picture I drew. Just with like highlighters and a pen.
Thank you.
But I think it demonstrates.
They're just chemicals, that's all.
So a mycorrhizal network is when, and I think this happens in nature all the time, is when mycorrhiza colonizes 2 plants at the same time. And what that means, and what we've shown in just experimental systems, is that the plants can share defense chemicals. So we can block everything above and below ground, and we can just allow the mycorrhizas through using the H pot in a greenhouse. And what Melissa showed is a reduction in ascochyta blight in chickpea when chickpeas were connected through a mycorrhizal network. We actually tried this with flax as well, so we did like a chickpea, flax. It didn't matter what they were connected to, it mattered that they were connected. Sorry for creating that. Farming smarter. Thank you, Henry.
You're good.
You're controlling this wind,
Yeah.
quite well.
It's exciting.
Turn down the
Compost. She just didn't do her talk fast enough.
There you go.
Just kidding.
So I think it's kind of exciting to be thinking about mycorrhizas potentially as, in defense strategies essentially for plants. I mean, it's not, again, I wouldn't necessarily recommend this as like a strategy, I guess, for mitigating.
Holy grail.
But it's not the holy grail. We maybe thought it was at the beginning. But we are seeing that plants with mycorrhizas seem to be doing better in terms of being able to do things like share chemicals and share defense chemicals in this case, right? So there might be a way to get signals down more quickly through a mycorrhizal network if you have a good mycorrhizal network going. Is there any questions?
Wasn't there some work done by Eric Swanson at the University of Guelph on peas about 8-10 years ago on this same thing?
Quite possibly, I'm not aware of work, unfortunately.
So I do remember talking about the whole idea of plants talking to each other for defense purposes and everything else.
Yeah, it's been done in like, I think fava beans and tomatoes are some really good experiments.
I think if I remember correctly, it was field peas.
Okay. I mean, I believe that, so it depends as well, like which plant you're dealing with. But I would say definitively in tomato, fava bean, and I think chickpea we have some decent evidence as well. And then of course not every plant is mycorrhizal. So brassicas, for example, are not mycorrhizal. So canola, don't put mycorrhizas on canola.
So sorry, you used this tubing device to show it, but how did you measure it? Like, how did you know what was happening? Like, how did you listen to the plants?
How do I listen to the plants?
Yeah.
There's multiple ways. In this case, we did it really simply. So we just assumed that our block worked, right? So the hyphae and the roots are different sizes. So we used a size exclusion mesh, essentially.
Okay.
Right? So then we're looking at if there's roots and hyphae versus if there's just hyphae versus if there's no mesh, essentially like natural system, right? So we can parse that out. And then we put bags over the plants because they emit volatile organic compounds, right? When something eats them, they scream, right? And chemicals, essentially. So we covered that up so that they couldn't talk to each other through the air. So that the only mechanism would be through there, right? So this guy gets chewed on, or in ascophytoblaitis fungus, right? So we put the disease on, it responds, and then we see a defense response in this guy who could only be talking through the network. Do you see what I mean?
Measure the chemical response.
Not in Melissa's. We just looked at.
Okay.
But yeah, you can label it, and we're talking about doing that, but we haven't labeled it. So we don't know what chemical it is, basically.
Oh, okay.
At this point, we can guess based on other work, but we don't know definitively.
Google Translate.
It's the future of AI.
Yeah, come on up. Look at our winter wheat.
So, when you're doing these testings, and you said the different varieties, so do you mean like between a wheat, durum, spring wheat, barley, or do you mean like within the spring wheats, all the different varieties are all?
Yeah, I mean, I'm seeing differences between varieties, like of spring wheat, right? So, like, Chris Gannett's PhD thesis showed differences in, like, current varieties that are used. I don't remember which ones, right?
But would you see more in a spring wheat versus a durum, or is that just it's all a difference?
Yeah, I would say like genetic differences between varieties are enough.
Everything.
I think is kind of what I'm seeing with our work and with the literature as well.
That makes sense.
So, yeah, and we're doing that like winter wheat. We're working with the winter wheat breeder right now to look at current winter wheat varieties and the ones that he's developing as well, to see if we can develop with mycorrhiza, which would
Make
be
it
the
difficult because of
the
all
dream.
the changes of varieties. So by the time you get
Well, it takes like what he said, like 8 or 10 years to get a variety out. So that's
so
about
you got
the
8 or
timeline.
10 years. That's your deadline.
That's my deadline. Yeah. Call me in 8 years. I'll have something. Yeah, I mean it's not ready, right? Like unfortunately, unless you're trying to do something that's like regenerative and super, you know what I mean? Like
That makes sense.
then then that might be something to think about. But if you're conventional, then we don't have a bug in a drug. That's this stuff yet, right?
So do you know what it is in the mycorrhizas that's doing it?
I think it's in the mycorrhiza.
Yeah.
So, yes-ish. It's when the mycorrhiza colonizes, it uses the same molecular mechanism as diseases often would through the root. So we think it's a priming mechanism, right? So it enters through that pathway, silences the defense response, essentially. And so we think so that immune system has been primed already. This thing is more like plants talking to each other, like as a warning system potentially.
But what is the line of communication? I mean, behind it, but what's inside the mycorrhiza that's actually telling the other plant?
So it would be the defense chemicals produced by the plant that would enter into the hyphae. There's some really great work being done out of Europe where they're looking at how quickly things travel through the hyphae. So you can just see like it's streaming in real time, right? So it's just there's essentially cytoplasm like liquid that's just running, right? So if you've got enough water that the thing exists, it looks like it could be a pretty quick mechanism if you can imagine a water-soluble, like jasmonic acid, which is, a general defense chemical that just kind of flops in and rides.
Are they aseptate? Like
They are aseptate.
Okay, so
Yes.
just flows.
It just flows. Yeah. Yeah, it's one of the features is they're aseptate.
Can you define aseptate?
They don't have cells.
Like the septa are just the cross walls, so it has no cross walls.
No cross walls. It's just like a tube, right? Like the hyphae are just like a tube where things can just flow through. Yeah.
So are brassicas non-mycorrhiza, or we just haven't found the ones that match them?
Yeah, I know there's people have said, oh, well, we just haven't looked hard enough. But yeah, I think they're predominantly, they just don't.
They're not.
They've evolved to not make symbiosis with mycorrhizas.
My question was around that then. So when you do grow a brassica, are you actually decimating what's there, or is it just that they're not increasing in numbers during that 1 year that you are growing a brassica crop?
Yeah, good question. It probably depends what you start with, but I did I looked for mycorrhizas like over there by Jail Road, and it was a pea and mustard intercrop. And we found very little, like too little to complete our experiment, basically, or like to ask any good questions about the system. So my guess is bad, and that can be because of the environment too. Like we're in a very dry environment, right? Like I think that selects against, and it was a dry land as well. So irrigation might help, right? Like I think maybe we're also in a environment that isn't very conducive to it, right? So, and this would have been 2019 or 2020, I think, experiments, whatever that year was, in terms of weather. But yeah, that was, I wasn't expecting that. I guess I'm telling you that because I was expecting that because there was like pea in there, which is mycorrhizal, that it would have just been reduced and not like.
There isn't. What about flax? Is that the same?
So mycorrhizal.
Really?
Oh, just what?
That's a good one. Flax is very mycorrhizal.
So
So there's also degrees, I guess, right? Like with
Yeah.
wheat, it's a bit like it's a average, I guess. Like it's mycorrhizal, but something like alfalfa is just goes bananas, right? Like mycorrhizal love it.
Are we supposed to be buying inoculant flat?
Unless it's naturally.
Am I understanding?
Well, it's also naturally available for the most part, right? Unless you've.
so you think there is a place that for some of this, the retail, some of this stuff is being sold, this mycorrhizal product?
Yeah, I mean, you can see like in Chris's work, right?
There is.
It works. Yeah. But he reduced, like, where it worked was where he did half the recommended rate of nutrients and then 1x of the inoculants, right?
Replacing.
Right.
So basically, and he got, I would say, like, I think it was pretty close to a 1x, right? So if you, I mean, pick your poison, right? Like or pick what you want to apply, I guess, right? In that case.
Spend more money on fertilizer.
So do you want to spend, I guess it depends how much fertilizer is, right? Like, that might be the calculation.
The pesticide, the fungicide, the herbicide that we're spraying, what effect does that have? Mycorrhizae.
Fungicides are pretty negative. But interestingly, we are still finding mycorrhizas when we spray fungicide, like foliar, right? We're still seeing it. So I think it's been shown in the literature that it reduces it a bit, but I think in our work, we still see that it's still mycorrhizal. So I think it probably
They're deep enough in the soil zone that you're.
yeah, where it just degrades and doesn't like affect in the soil
But
as
then what
much.
about fungicide seed treatments?
Yeah.
Yeah,
Because that's right on the roots.
yeah.
But how far does that spread out into the soil?
Good point.
And how long
Yeah.
does it last?
Yeah, I haven't seen work on seed treatment. I'd like to do that, but
What if it's not a fungicide? What if it's a herbicide with surfactants and adjuvants and everything else?
Residual
How-
herbicide. Yeah.
well, like if you're using a herbicide, like you're drying out your plants, right? So you're essentially killing off their carbon source
Right.
because they're an obligate. They get their carbon from the plant. So what you're doing in that case is probably triggering sporulation. They go dormant at that point.
But that's it. You're only killing hopefully only killing the weeds, not the actual crop, right? So the
Correct.
mycorrhizal the crop will still.
Well, you're not gonna kill the mycorrhiza, right? But you're gonna shorten their lifespan, right? Because you're shortening the lifespan
The weeds.
of the weeds and of the plants,
Right. So
right?
it's to do with the host, not the actual product itself. On
Some of the weeds are mycorrhizal.
Yes.
Oh, yeah.
It would be, yeah, for sure. We had a section of flax this year, half the seed on mustard, half on lentil ground. And the flax on lentils looks amazing, the flax on mustard looks terrible.
Yeah.
But that's what they're doing studies on, right? Is that mustard is a fumigant. So
Yeah.
Yeah,
Just
well, it's allelopathic
mycorrhizal.
too, right? Like
Same
fumigant,
goes on.
exactly.
Bambi or
So
that.
they could be other things, not just
flax-
this.
well, canola between male and female.
Your lentils also really good nitrogen.
What's that?
Yeah.
Yeah, it could be a number.
The number of things.
It could help, I guess.
It makes you never want to put
Hey,
plants
Monica,
on
do you mind repeating the question just so everybody can participate in the
Sorry.
conversation?
Thank you. So the question is, if I'm intercropping with chickpea and flax, do I add an inoculant? I think I've seen studies, I guess unpublished, I think still, that just intercropping with flax is really good for reducing like blight, ascophyta blight. So maybe not necessarily, but yeah, I mean, that was kind of the question we were trying to get at. So I don't know if I would say inoculate. And I hesitate to say inoculate or add inoculum because the literature isn't clear, right? Like whether it's actually going to be helpful or not. So again, I would say hold off until we discover the right mycorrhiza for your chickpea and for your flax that you're planting. Because I'd be afraid that it won't work because basically you're taking a gamble, it's like 50%. Half the time it doesn't work, that's what the literature says. Or half the time you don't get the yield boost that you're expecting.
So, is that what you're seeing between male and female bays? Is that a dumb question?
Sorry,
between male and female bays, like when you grow a crop after a seed canola, a lot of it has to do with trash. But I wonder often if, like, a crop always seems set back in the male bays where it was male bays last year.
I have no idea.
For seed canola?
Mm-hmm.
I thought it was the other way around.
What's that?
I thought it was the other way around. The male was better.
This is a canola question. It's a non-mycorrhizal plant, so I know nothing about it.
Gender bias? You're not bringing that conversation up.
just because you're mowing out the males.
No, I honestly, I don't know. I don't know canola production very well, so I don't know.
Either. There's a lot of questions.
Yeah.
Monica, there was a paper recently that said something about canola non-mycorrhizal, and that the relationship wasn't so much that it was non-mycorrhizal, except that maybe it's the lack of available phosphorus. So what is the phosphorus dynamic, the phosphoric amounts? How does that impact the mycorrhizal populations?
Like whether there's phosphorus available in the soil.
Yeah, because I mean the
Okay.
Whenever you have a crop after a non-mycorrhizal crop, the issue is that the next crop isn't able to access early phosphorus because the mycorrhizal populations are too low and the plant isn't really good at getting it. So I think the new paper said something along the lines that it's not necessarily that it was a non-mycorrhizal crop, except that the available phosphorus was lower, so the mycorrhiza couldn't do its job.
Right.
Did you see that one?
I have not seen that one, so I don't know.
But Ken, from my experience, I've seen that situation where even where you still have that purpling, that phosphorus deficiency, even on very, very high testing soils. So I don't think it's that's unavailable. It's for some reason they can't take it up.
Well, that's kind of what available means, buddy.
I said no. Depending what gender you are, according to Jenny.
Not talking about that.
I feel like it's also plant available, right? And I think what we know is that mycorrhiza partner with phosphate solubilizing bacteria, right? So they don't pick up the phosphorus themselves. They pick up the bacteria that do the thing and then suck it up from, so they're going further and deeper, so they're able to access more, I guess, volume, you could say, right? And then they're able to partner, or they bring their own bacteria, essentially. A lot of the strains don't function well without, like, endobacteria that live within them, essentially, that they deploy to do that. So I think, my guess would be that having a mycorrhizal plant at that point would help you access that phosphorus, but...
That's a better way of putting it. It's good. Ability to access is probably a better way of putting it.
Yeah, because it can be unavailable to the plant, right? So, yeah. Yes,
Another
Tom?
thing too, the brassicas exude types of organic acids that naturally solubilize the phosphorus. So they take the less soluble phosphorus, make it more available to the plant. So they have a different mechanism to get
So
it.
they don't need the mycorrhiza. They just
Yeah.
need to,
Yeah.
Okay.
Because they do it differently.
That's one theory.
I'll buy that.
I believe in fertilizer. Plants don't have the mechanism to be affected by mycorrhizas.
Yeah, I mean, like mycorrhizas. The theory is that they are that they helped plants colonize land like 400 million years ago during evolution, essentially. So most plants have it, but then I guess brassicas lost or figured out a different way, right? There's also like plants in what's that group in Australia, basically, because they have really depauperate soils in Australia, really old soils, right? And so they use like a whole different mechanism to get phosphorus out. That isn't mycorrhizal at all, right? But it's just like a solubilizing. Yeah, I'm sorry, I don't know the mechanism very well, but I know that that's one of the examples of a group that's evolved to exist in a place with lack of phosphorus, essentially. I need a drink. Any other questions over here? Everybody's happy? Okay, if there's no more comments, we can wrap it up and send you to Henry's side.
Is The Last of Us possible?
Haven't watched it.
Oh my god.
I have.
That's supportive.
Okay, ask her. She's watched it and she's a mycorrhizal expert.
It was filmed here in Lethbridge.
There was images of it in my bio class and from the previous instructor. I had to look up what the hell they were because I hadn't watched the movie.
Now I gotta watch it again.
Yeah.
Well, it's a cordyceps
Mycorrhiza
fungi
and
that
cordyceps.
jumps to people.
Oh.
So that's what
Then it starts
What's the
controlling
difference
people.
between mycorrhiza and cordyceps?
it does.
Yeah.
Cordyceps is a saprotope. Well, no, cordyceps infects insects.
We need a party.
And kills insects and grows out their
But now we
eyeballs.
can't watch
Oh,
it.
Right?
Yeah.
But mycorrhiza forms a benevolent symbiosis with the roots of plants that
Okay.
helps them grow.
I'm gonna
So
go with
one's a
boss.
pathogen to insects.
I think it's possible.
Yeah.
One depends on the life of a plant to proliferate itself. So just different mechanisms, right?
Really popular.
Yeah. So
I mean, cordyceps
kids
is a
afraid of nothing.
some, right? I guess which is what the movie's based about then? Is that-
It's a
okay.
show. It's
Okay.
based on a video game that's based on
Thanks.
some
HBO.
Sorry, I
protruded
exited pop culture when I
fungi.
had kids 7 years ago and haven't returned yet, so.
They might get you back into it. But
I mean, I'd like to come back, but I'm not here yet.
You're not there, you go.
Yeah.
Thank you very much, Monica.
Yeah.
Thanks, Monica.
Yes, thanks, Mom.